Okay, let's now compare the physiology of voluntary striated muscle to the only involuntary striated muscle in the body, the heart. Like skeletal muscle, you can see the characteristic striations or striped patterns along these short, fat, branched muscle fibers that are so characteristic of cardiac tissue. This picture is from a 100x magnification under your microscopes in the lab. Troponin and tropomyosin still regulate contractions, just like we talked about with skeletal muscle. But like smooth muscle, the pacemaker potentials, which are slow, sustained depolarizations, are due to the intracellular fluid increasing in sodium and calcium working together. That would be during the rising phase that you would see on an action potential. Both ions work together, which is unique in the heart. Also unique is the pacemaker signals that occur without any extrinsic neural input from the central nervous system, and this is due to gap junctions that help transmit electrical signals that originate way up in the right atrium from the sinoatrial node out to the entire heart, cell by cell by cell, and we'll look at those details here in just a second. So now that you guys have all had a chance to look at EKGs and do the study questions and look at those supplemental videos, I wanted to circle back around and discuss heart muscle function a little bit with you as it relates to the electrical pacemaking abilities that we've looked at with the EKG patterning. If you think about the EKG and what you've learned, and then consider this picture from your textbook. This is 1311. What we want to do as we finish up our studies on EKG today, let's look at what's happening in the heart as a whole due to that rhythmic depolarization process set by the pacemaker. So in this figure, you want to start at the top, up at 12 o'clock, and it's labeled panel A. And then we're just going to go clockwise around this diagram. And what we get into here is following that yellow highlighted area. That's indicating the part of the heart that's being depolarized as we go around this diagram clockwise. So the structures to consider would be the pacemaker up at the top, the right atrium, the sinoatrial node, and the AV node, so there is some redundancy here, the bundle of His, and then the Purkinje fibers that branch and take a U-turn down at the apex. They're highlighted in green, and they go up the walls of both ventricles. So that's our electrical pathway. And if we start going around this diagram, panel B, we see a yellow highlighted zone up at the very top of that right atrium. So the sinoatrial node, or our pacemaker, is depolarizing first. So that sets the pace of the heart. So your cardiac cycle going from contraction to relaxation is going to be driven by these rhythmic depolarizations, and those are indicated by the yellow zones on this diagram. So from the depolarization of the pacemaker first, um, because of proximity, the atria will depolarize slightly ahead of the ventricles. So the top two chambers in panel C are both highlighted yellow because the SA node and AV node are now conducting, and those atria here on the top are going to depolarize first, and which is good because they're going to contract following depolarization, and the blood that's in the atria during contraction is going to be squeezed and forced through the AV valves into the ventricle. So that's how we move the blood through the heart. So if we come down here at 6 o'clock, notice the atria are no longer highlighted in yellow. It is now the job of the Purkinje fibers to carry the depolarization signal down to the ventricles. So in panel D, the only part that's highlighted is the septum, kind of the wall that separates right and left heart. And the Purkinje fibers use that as a little freeway, and they're depolarizing. And if you come up here to panel E, you see the ventricles are fully highlighted in yellow, and they're undergoing depolarization. And then up at the panel F, almost back up to the top, we see that there is no highlighted yellow anywhere. So just like we mentioned with skeletal muscle, relaxation phase is just as important as depolarization. So the entire heart here in panel F, up here on the left, at about 11 o'clock, it's going to be returning to its resting state, and this is one cardiac cycle, but we're just considering electrical excitation and relaxation. Okay, so if we zoom back into the cells, okay, so we've looked at organ level, tissue level. Let's look at what's going on back in the cells. If we zoom back in, we're once again faced with having to understand the steps involved with excitation, contraction, coupling. So this diagram should look familiar. Let's go over the major differences, however, when we compare skeletal muscle excitation, contraction, coupling to the same process in the heart cells. So we know that the pacemaker is going to set the rate of depolarization and the consequential contraction in the heart. But the differences that we see when we look at this similar looking diagram is, first, there's a lack of external nerve stimulation, so you don't see any synapse up at the top with the somatic motor neuron, because this is involuntary muscle. So we just want to remember that the pacemaker is setting the rate of depolarization. So it's internal, it's from within the organ itself, no external stimulation. And then we also see that the cells that make up cardiac tissue are all connected to each other. And this is a direct connection through the gap junctions between each cell. This structural feature means that the ICF compartment of every cell is connected to its neighbor, and what happens to one cell happens to the other. And then also, if you look along the sarcolemma up here at the top, we can see a lot of voltage operated channels. Okay, so you're going to have a lot of voltage operated channels along the plasma membrane or sarcolemma. So when you get a depolarization, what happens is you're going to see an influx of calcium and sodium working together. Both ions will rush into the cells. You'll also see a loss of potassium, and all of these factors are going to contribute to the rising phase during depolarization that you would see, in any other action potential graph. So consider that the sarcoplasmic reticulum is still very well developed in these types of cells, just like we saw in skeletal muscle. The SR is going to contribute to the elevated calcium levels inside the cells. So for the first time, we're noticing what's called a calcium-induced calcium release. And this is what's going to allow for that coordinated contraction, relaxation, phasing of the heart that's so characteristic. So the two ions work together for the rising phase. You'll need to review this diagram. It's numbered for you. So I just wanted to point out a few differences, but you still see a lot of the same structures that we studied for skeletal muscle, and you'll want to definitely study this diagram before the exam on Monday.